Hello and welcome to the second part of the trilogy, Let's Make Some Sugar. This is the light-dependent reactions, Part 2. In this video, we are going to talk about the first component of photosynthesis, the light-dependent reactions. This is what photosynthesis actually is. Now, let us define the light-dependent reactions. This is a set of several redox reactions which involves the transfer of electrons down the increasing reduction potentials of redox carriers or the QUANTASOMES. This reaction or these reactions uses water and light to generate ATP and NADPH as energy-rich molecules by expelling out oxygen as a byproduct. Now, again, a question similar to the previous that we have asked arises here. That where do you think these light-dependent reactions occur? Whether it occurs in the stroma or the lumen. It doesn't occur either in the stroma or in the lumen. This process occurs in the thylakoidal membrane because the thylakoidal membrane consists of QUANTASOMES. This is the point where we will talk about the QUANTASOMES in detail. So let us talk about them now. The QUANTASOMES. These are the photosynthetic machinery of a green plant. These QUANTASOMES are particles found in the thylakoidal membrane of chloroplasts in which the photosynthesis actually takes place. These are composed of lipids and proteins that include various photosynthetic pigments and redox carriers. So, a light-dependent reaction uses water, light to generate energy in the form of ATP and NADPH with evolving oxygen as a byproduct. Let's dive into the details of this process. The light-dependent reaction uses water and energy from a photon of light to build the molecules ATP and NADPH. ATP and NADPH are used as fuel for light-independent reactions later. Just a minute ago, we talked about the QUANTASOMES, which are arranged according to their increasing reduction potentials in the thylakoidal membrane. Now let us talk about that what is actually happening in these QUANTASOMES. Let us talk about the photosystem 2 first. From our earlier videos, we know that what is a photosystem. This will be a good idea to go there and watch that video and come back here to understand the things better. In order to explain the things in a simpler way, I will again take an illustration or illustrations from Jay Hosler's blog. Let me introduce the characters once again. Here is Aunt Edna and Wilbur the hoverfly. As shown in this figure, that photosystem 2 consists of antennal complex and a reaction center. Aunt Edna explains that let us consider this as a chlorophyll molecule with a head and a tail, and he designated Wilbur the hoverfly as the reaction center P680. In the second illustration, Aunt Edna said that if we consider our bodies as chlorophylls, then our heads will act as electrons. And if we flash the light on any pigments contained in the antennal complexes, their electrons get excited. We have talked about these things in our earlier video on the photosystems and the pigment systems. These electrons got excited to a higher energy level. And in the third representation in Jay Hosler's blog, he explains that how these electrons, which are in the excited state, passes its energy to its neighboring pigments. And finally, this energy is transferred to the reaction center. And obviously, it will excite its electrons too. So, this is what happens in a photosystem 2. Finally, the energy got to the photosystem 2 by resonance energy transfer, and it excites its electron, which will be snatched by a molecule, the plastoquinone. If you pay attention, you will notice that P680
aka Wilbur the hoverfly, lost his head, that means the electron. And now the question arises that from where it gets the electron back or his head back. The answer is it will get its electron by the functioning of OEC, oxygen evolving complex. The oxygen evolving complex split the water molecule to replace the electrons lost by the photosystem 2. The OEC will split H2O molecule and it will take its electrons to PS2, releases the protons in the lumen and cranks out the oxygen molecules during this process. That means finally Wilbur got his head back. That means PS2 is replenished its electron. Oxygen is evolved in this process with protons thrown into the lumen. Oxygen will diffuse out from the lumen because it is a gas. So oxygen is evolved in this process. Now the electrons that are taken in by the plastoquinone, their number is 4 because of the splitting of 2 water molecules by 4 photons. Now these 4 electrons are taken up by the plastoquinone, and plastoquinone will provide these electrons to the next complex with an higher reduction potential among the quantasomes. The next complex among these quantasomes is cytochrome complex, also known as cytochrome B6F. Now, this cytochrome has a specific function. Its specific function is to pump protons from the stroma into the lumen. Therefore, 4 electrons will pass across the cytochrome complex, they will pump 8 protons into the lumen. Furthermore, these electrons are transferred to its next destination via a different type of electron carrier. And that electron carrier obviously has a reduction potential greater than the previous one. This is named as PC, which stands for plastocyanin. The plastocyanin will take these electrons and transfer them to the photosystem. This is also powered by 4 photons. Once again, here no electrons are snatched from PS1. It just acts as a bridge that will transfer these electrons that actually or originally came from photosystem 2, and it will transfer these electrons further via next redox carrier, ferritoxin. And ferritoxin will provide these electrons to NADP positive reductase. The NADP positive reductase uses these electrons to reduce NADP positive and generate NADPH within the stroma. So we have generated NADPH. Now this is the time to generate ATP, and for that we must go back to our previous quantasomes. Here we can see that there are 4 protons that are dumped inside the lumen by splitting of water by OEC. Secondly, there are 8 protons that are pumped inside the lumen. There are considerable number of protons inside the lumen already, but by transferring these electrons, we generate an electrochemical gradient. This is a preparatory mechanism to generate ATPs. Now, how ATPs are generated? To understand that, let's go to the next slide. Electrons are taken from oxygen evolving complex by splitting of H2O, taken by plastoquinone, transferred to cytochrome complex. Due to the transfer of electron among the quantasomes, there are several protons that are pumped inside the lumen of the thylakoid. As you can see, actually there were 2 ways to enter protons inside the lumen. First, by splitting by oxygen evolving complex, and second, by pumping protons by every electron that passes through the cytochrome complex. But in order to balance this electrochemical gradient generated, or in order to escape out, the protons do not have any path except ATP synthase. ATP synthase is an intramembrane structure that looks like a mushroom. We will talk about its general structure in our upcoming future videos. This is the only pathway from where the protons can exit. Protons will exit through this, and during their exit, they help in the formation of ATP. If you will notice carefully, it is happening in the stroma. So, now our goal to generate ATP adenosine triphosphate and NADPH or energy is completed. So come, let's summarize what we have studied so far. In this picture, I am drawing thylakoidal membrane with lumen and stroma. First, 4 photons strikes photosystem 2 and excites its electrons to a higher energy level. These electrons are carried by plastoquinone one by one, not simultaneously, to cytochrome B6F. The electrons lost by PS2 are replenished by splitting of 2 water molecules into oxygen and protons and electrons by oxygen evolving complex. While these electrons are transferring from cytochrome B6F, 8 protons are pumped inside the lumen. Now, the plastocyanine will take electrons from cytochrome B6F and replenishes the electrons that are lost by PS1 to ferredoxine when 4 photons strike the same. Now, these electrons are eventually transferred to FNR, which is an enzyme that will reduce the NADP positive to NADPH. Now, NADPH, the first energy molecule, is found. Now, this is time to produce ATP. As we know, that splitting of 2 molecules of H2O puts 4 protons in the lumen. Similarly, the transfer of electrons also dumps 8 protons inside the lumen. This increases the concentration of protons inside the lumen and make the lumen more positive inside as compared to outside. There is no way to get out from the lumen except one, the ATP synthase. This is an inverted mushroom-like structure that I have shown in this picture. It allows the protons to come out, and while doing so, it generates ATP from ADP into the stroma. Both ATPs and NADPH are found into the stroma. This was all about the things that are happening inside the thylakoid, summarized in everything that is happening in the thylakoid that powers the formation of NADPH and ATP is basically the electron flow. This electron flow may be of 2 types, cyclic electron flow and non-cyclic electron flow. Let's talk about them also. First, the cyclic electron flow. Cyclic electron flow involves only PS1 but not PS2. In this case, the electrons are flown from P700 of PS1 and move through B6F complex back to P700. The cyclic electron flow is also known as the cyclic photophosphorylation. There is no production of NADPH and no release of oxygen in cyclic electron flow. Similarly, on the other hand, in non-cyclic electron flow, there is involvement of both the photosystems, photosystem 1 and photosystem 2. This is also known as non-cyclic photophosphorylation. And in this process, NADPH with ATP is formed, and also oxygen is evolved because of the presence of oxygen evolving complex. Both NADPH and ATP are found in the stroma. As the electron flow is not cyclic, but it forms a Z shape. That's why it is also known as the Z scheme or the Z electron flow. That is all about this video. In our next video, we will talk about the second component of photosynthesis, the dark reactions or the light independent reactions. The next one will be an interesting one. Stay tuned and like, share and subscribe my channel. Thanks for watching.